do is give a bit of an insight or a glimpse of the kind of experiments we're doing in my group in Delft, and also put it a little bit in the context of this larger ambition of building. Of course, is a, who knows, it's a dot on the horizon. It requires a big effort by many people, but we, like you said, we started a national program 3 years ago now, between physicists, biologists, chemists, to make a first step. Of course, the program is about building a cell, but I imagine that will take a bit longer depending on how you define success in such a project. Anyway, this is a slide showing the group as it's currently composed. So we're at the Department of Bio-Nanoscience at the Technical University of Delft. So this is a physics-based department that now exists about 10 groups, junior assistant professors, associate full professors, independent groups with a background in physics often, but also in chemistry and biology, where we really look at very fundamental way in how life works, let's say, from the molecular scale to the cell up. So it's quantitative cell biology and nanotechnology, single molecule biophysics, and also theory combined in this department. So it's a huge pleasure. And my group currently consists of people that are listed on this slide. So I have PhD students. Actually, often they are co-supervised with other groups in collaborative projects, postdocs and technicians that provide biochemical support and microscopy support, also often shared with other people. So this is roughly the group size that I'm happy with. And then I list names former PhD students and postdocs in my lab. There are, of course, many more, but that I show today really to their credit as well. So the Department of Bio-Nanoscience, maybe is nice to mention, is also part of the Kavli Institute for Nanoscience that we have in Delft. And this includes a second department for quantum nanoscience. So it's at the similar scale, but with very different applications in mind, like quantum computers. So the title that I chose is Reconstituting Cytoskeletal Systems in Artificial Cells. So I will give some examples later on, but we are inspired by biological cells, motivated also by understanding how cells work with a focus on the cytoskeleton. So the cytoskeleton, and I will introduce it in more detail later on, is, let's say, the mechanical framework of all eukaryotic cells, and actually also prokaryotic cells like bacteria have cytoskeletal systems. And it provides for mechanically stiff components that together can organize in different forms depending on what the cell needs to do. And of course, there's a lot of cell biological research on how the cytoskeleton works in cells. But our contribution, let's say, to this field is to try to build from the bottom up in artificial cells, and I will explain what we mean by them, minimal systems of these cytoskeletal networks in an effort to try to understand mostly the mechanical side of it. So these are dynamic force generating filaments, as I will show, that self-organize, for example, to organize chromosomes during cell division. And by building from the bottom up, we try to understand or contribute to the understanding of how these structures work. So this is just some examples of these artificial cells. The examples shown in this first slide are all microfluidic droplets that are made using microfluidic techniques that provide a closed-off environment roughly with the volume of cells. So the diameter of these structures is typically 10-20 micrometers, like you might expect for many eukaryotic cells. And then we enclose in these artificial cells proteins that are purified from either from bacteria or from tissues or expressed and then purified from cultured cells, and then engineer the content of these artificial cells with enough components that we can study some of these self-organizational principles. And I'll get back to the details in a second. So as I promised, I'll put it a little bit depending on how much time I have in effort, and this is this building synthetic cells. And I'm just gonna slow 2 slides about it now and then maybe come back to it at the end. So, you know, if you think about this approach to understanding biology, trying to build bottom up from known components or a minimal set of components on biological functionality. You could dream and say, okay, can we go all the way? Yeah, of course, we are not the only ones doing this around the world. There's lots of groups in Europe, in the US, in Asia, actually everywhere, that have built over the years this capability of reconstituting cellular functions outside the cell. And you could ask yourself, okay, is there a way that we can bring enough of these cellular modules, as we call them, together, such in an artificial lipid container, for example, such that we can reproduce all the essential functions that cells have. And then, of course, you can have long discussions on what those functions could be. But for us, we are looking to build a membrane-contained system that has enough autonomous components that it's at least able to grow and divide and transmit information in the form of DNA. So that's what this project is about. And so then you might ask, okay, why do you want to build a synthetic cell? And I think there's two answers to that. First of all, you could think in terms of applications. So of course, we use real cells and cell engineering already, for example, to use yeast and E. coli cells to make things for us as factories. But if you are able to bottom up build these cell factories, then you can maybe make them more specialized and then find new production methods for maybe materials, medical molecules that can play a role in medicine, but also biofuels. So it could be adding to the biotechnology that we have, but a bit more simpler engineered systems. That's not the consortium in the Netherlands that we have is not necessarily aiming for these applications immediately, because there's also just a fundamental curiosity side to this, and I can maybe explain with these slides. So you may have all seen these, this movie on the left. It's from The Inner Life of the Cell, and it's an artist's impression of what you may see when you make yourself very small inside a cell and look at how transport takes place. So what you see here in this artist's impression is a kinesin, a molecular motor. It's actually two proteins together forming a motor, a dimer that hooks up to a vesicle, a membrane-based container with cell components that gets transported in a directional fashion through the cell. And we all know that the cell, which has DNA codes for all these proteins, then builds all these proteins, and the collection of not only molecular motors, but everything else that is there in the cell somehow gets organized in time and space in such a way that life emerges. And I think the field, biology, biophysics, has really progressed in the last decades in understanding these individual cellular components. So kinesin is a nice example where really nanotechnology has helped, even already in the late last century, to study how such a single molecule works. How does it walk? How much energy does it burn? What does it depend on? What are the forces that can be generated? And we have a lot of information about many other cellular molecular components as well. But I think it's also still fair to say that we don't know, we don't have a real understanding of how we need to put all these molecules together in space and time to make something possible that you see on the right, which is an example of what all life can do. This is a fission yeast cell, a single cell organism, where the DNA, the genetic material, has been fluorescently labeled in green. And you see in this beautiful image, which comes from a Japanese lab from Professor Niki, that cells have the autonomous ability to grow twice as big and at the same time duplicate exactly once their DNA, then separate this DNA faithfully to two sides of the elongated cell, divide in the middle, and thereby create two copies of itself. So that's all our cells, all cells have this basic capability of reproducing a copy of itself and do this multiple times. So the big question, fundamental science question, is how do you go from knowledge about individual components to understanding how this all cooperates in a cell that is alive. And the idea being that, of course, we can study these cells, and we should, but by building from the bottom up, piece by piece, the elements that make this possible, we hope through this project to really gain fundamental understanding of the working principles of living cells, in addition to hopefully being able to also use these mimics of living cells in the future. And if I don't get time to say it later, I don't think the goal is to really reproduce cells as we know them around us, because they're probably way more sophisticated and you know, and optimized than anything that we will be able to build bottom up. So it's not the idea to reproduce exactly what already exists in nature, but to make, I sometimes call it a flight simulator, minimal versions of them or mimics of them that give us a tool to really understand, control, turn knobs, and understand how cellular processes work in the context of a complete cell. And as already mentioned, you can take also a top-down approach and ask the same question. Take a very simple cell, reduce its number of components by taking out pieces of the DNA, and ask the question, what is minimally required to be alive? And that's a complementary approach. And I think we will be in good shape once these two approaches meet each other sometime in the future. Anyway, that was a long story about the context. I want to go back to what we are doing. So we're interested since many years in the microtubule cytoskeleton, and I'll give sort of talk you through the kind of experimental or experiments we've been developing over the years to understand better how the cytoskeleton gets organized. And as I already said, we are inspired by what happens in real cells. So you see here two examples of these. What the microtubule cytoskeleton, I should say, is responsible for in real cells. One is cell division. So you see two versions, one movie and one picture of a cell dividing. The one on the left is a cell in a dish sitting on a flat surface, where you see very nicely the chromosomes in red being separated to two daughter cells. And the mitotic spindle, which is a self-organized structure based on microtubules, is every time the cell divides, self-organizes to be able to do this extremely faithfully. And the picture on the right is an embryonic cell, so it's a first cell stage, which also shows very beautifully that not only does this structure divide the chromosomes to two little cells, but it also knows where it is with respect to the confined space of the cell. It really positions, in this case, along the long axis, slightly asymmetrically, in order to make sure that when this cell divides, it divides along the correct axis, and also makes two slightly different size cells, which is important for embryonic development. So what on the right, you see that this same microtubule cytoskeleton in a different context can perform a very different function. So this is again the fission yeast cell that I also showed in the synthetic cell context, but now highlighting the microtubule cytoskeleton in these cells when they are growing. So this is not a dividing cell, not at the moment we take this picture at least. And on the bottom, you see how an anti-parallel array of the same microtubules that you see in green on the left, in this case, is responsible for bringing cell end markers to the cell end. So you see these green dots at the cell ends. They are molecules that need to be at the cell ends to make sure that the cell grows in a linear fashion and not sideways. And if you look on the top, you see how these dots, these green dots that are white in the movie, actually get brought to the ends of the cells, not by diffusive processes, but by associating with the dynamic filaments, the microtubules that bring these molecules towards the cell end, where they then hook up to a receptor molecule that is asymmetrically distributed. So in both of these examples, what we are asking is, can we build a minimal version that reproduces the sort of the fundamental aspect of what these structures are responsible for doing in living cells. So the one on the left is, and I'll give you more detail, is our efforts in building minimal mitotic spindles, where we ask, what does it take to get this typical bipolar organization? What is the force balance in such a structure, and how is it then affected by all these other molecules that we know play an important role in real cells? Because you have to realize, no matter how similar or not, depending on how you look at it, our spindles look to the real thing. Of course, they're infinitely simplified compared to the real thing, which will always be way more beautiful. So on the right, we have a similar example where we ask, we know that this pattern, if you want, of cell end markers is dependent on a dynamic cytoskeleton. So what does it take minimally to create an asymmetric pattern like this with the help of linear transport by microtubules? There are other examples in nature, for example, in E. coli cells, where these asymmetric protein patterns are set up by reaction diffusion mechanisms. In the case of E. coli, it's a MIN-CD system that actually oscillates. It doesn't give a steady pattern, but an oscillating pattern. So we have quite a bit of understanding of how reaction diffusion can set up these mechanisms, but we're interested in understanding how a combination of linear transport and presumably also diffusive processes can also set up these patterns, and what are the properties of these patterns. So by the way, I know we have time at the end for questions. I do not see most of you, but if you're if you're interested in asking a question in the middle, that's also fine. So a little bit of background for those of you who do not know about microtubules. This is again an artist's impression of what a single microtubule looks like. So it's actually a hollow tube built of proteins. So there's protein subunits, actually dimers, that self-assemble inside the cell or outside the cell into hollow structures that have 13 what we call protofilaments, and assembly takes place at the end. So that's what you see happening here. There's an energy consumption associated with this growth process, and then every now and then this elongating filament switches to a shrinking state. So the energy consumption is a very important element of that ability to switch between growing and shrinking states that I will not go into too much. But the end result is that you have fairly stiff rods. It's a 25 nanometer diameter rod that has a persistence length of millimeters, which means it's basically a stick, a rigid stick that can grow, reach all the way to the other end of the cell, push on things that it encounters when it's growing, that can then completely disassemble again by switching to a shrinking state, and thereby has a lot of flexibility in reorganizing and reinventing itself all the time. And what's interesting to us that the growing filaments can generate pushing forces, but also when the filaments shrink, if they hook up to something that, for example, a chromosome during cell division, they can also exert pulling forces and thereby really transport stuff through. So these dynamic filaments, we've been studying them outside the cell. So I'll show you now a series of movies of the kind of experiments we do to better understand these filaments. And we're not the only ones, obviously. So each time on the top, you see the behavior that we were reconstituting in a real cell. So this is, for example, a movie made by Anna Akmanova. She's a cell biologist in Utrecht with whom we collaborate a lot. And this is not an artist impression, but really a microscopy movie where you see individual microtubules, 25 nanometer in diameter. You don't resolve them, but you see them in this fluorescence image, which has then been, changed in color, but anyway, in contrast. But if you look carefully, you really see this switching behavior between growth and shrinkage. And on the bottom, you see that behavior reconstituted purely based on tubulin, the subunit of the microtubule. So we take a glass cover slip, we stick a piece of microtubule that has been color labeled in pink, chemically labeled so that we can stick it to the glass, and then we provide the system with energy molecules, GTP and tubulin dimers, and you reproduce this growing and shrinking behavior, including the switching between growing and shrinking, without anything else, showing that it's really intrinsic to the polymerization behavior of these proteins. So we can study very precisely, and this has been discovered already in the 80s by Tim Mitchison and Mark Kirshner, and the microscopy has improved, but the principle already was known many years ago. You can study how this depends on the concentration, temperature, anything you want. It's very well controlled. So our focus has been on mostly over the years on the forces that can be generated by these dynamic filaments. So and that's what you see here. So we can repeat that same experiment, but now we do this in front of an artificial barrier. So in these movies, it's an two microscopy versions, one old from when I was just starting my group and one more recent, where we present this dynamic filament with a microfabricated barrier made in the clean room. And then you see in these looping movies that when a growing filament hits a barrier, when it's stuck to the glass on the left, it grows into this barrier on the right. You see that it actually continues to grow, and a force is being generated by this growth process, which then leads to bending of the filament. And since we know the mechanical properties, the bending rigidity or the stiffness of these filaments, we can measure them separately, independently. We can study this buckling behavior and deduce from this what the forces are that are generated by these growing filaments under different conditions. And we can then also ask, okay, if you put a force on the end of a growing filament, how does it affect the growth velocity? How does it affect the probability to switch from growth to shrinkage? We can connect the results to thermal redshift models that give predictions about these force velocity behavior. So there's really a lot we can learn about the biophysics, and we've done that in the past. And just to show that this matters also in cells, I show on the top again a fission yeast movie where these dynamic microtubules are growing from a site where they are attached to the nucleus. You don't see this nuclear membrane here, but the filaments grow from the nuclear membranes towards the cell end, where they display the same behavior. They bump into the cell end, continue to grow, buckle, and then switch to shrinkage. And these forces that are generated at the cell ends this way keep the nucleus in the middle of the cell while the cell is growing. An important observation is that when you generate a force like this at the growing end, the probability to switch from growth to shrinkage becomes much higher. And that sort of self-organizes the fact that these filaments never grow too long, because when they reach the end of the cell, they have an enhanced probability to switch to a shrinking state, which automatically tunes the length of these filaments to the size of the container, which in this case is the cell. So then we are interested in how do these dynamic and force generating capabilities contribute to self-organization of a spindle inside a confined space. And I should also say that I'm not showing it here, but we can also measure the shrinking forces. So instead of a passive barrier, you can also put a connector molecule at these barriers, like a molecular motor that is able to fix itself also to a shrinking filament. And it's all published if you want to know more details. And then by putting the left end, which is in this movie stuck to the glass, to a bead, for example, in an optical tweezer, with a more sophisticated experiment, you can also measure the pulling forces that are generated by shrinking filaments. And then we ask, how do these combination of pushing and pulling forces contribute to positioning? On the top is again the C. elegans embryo that I already showed. And we asked, for example, if you take half a spindle, a very minimal version of it, which is a radial array of dynamic microtubules that are able to interact with the boundary of a microfluidic container. And we put actually molecular motors there that can hook up to shrinking filaments. Then we can really study how a combination of pushing and pulling forces generated at the boundary of this spherical container leads to faithful positioning of this astral structure. And I will not get much into it, but most recently, we've also switched some of our attention to the biochemical regulation of all this, because even though mechanics is important, cells, of course, have lots of biochemistry going on. And again, you see here a movie of Anna's lab, which is highlighting a protein called EB1. It's an end-binding protein, actually EB3, sorry. And it's a molecule that specifically recognizes the growing end of dynamic microtubules, hitchhikes with this growing end, and provides a platform there for other molecules to land on. And these molecules, for example, allow the microtubule ends to hook up to chromosomes or the cell cortex, but also molecules that regulate the dynamic properties of these filaments can land there. So there's the mechanical side to it, but increasingly we're also adding control molecules that are known to be important in cells also to our system. And that's what you see in this movie. So it's a similar radial structure as the picture on the left, which in this case fails to position itself nicely in the middle in a way that we also understand. And that has these end tracking molecules added to it, which really make its filaments way more dynamic. And so depending on whether the filaments can actually robustly interact with the boundary or not, meaning depending on how often they switch between growing and shrinking, you can get different regimes where positioning positioning is possible or not. And that's something that we can tune with temperature and concentration, but we increasingly interested in tuning that with control molecules that also play a role in cells. So it's a long story about, I sometimes call this the story of my life. It's sort of over the years, the different kinds of techniques that we've developed with always the goal in mind to get to this, what we call functional complexity in vitro, getting more and more complexity in to become more relevant for what happens in real cells, but still simple enough that we can connect to theoretical models, really understand the mechanical things that are happening. So just, I will not spend too much time on, but just to show that we do this buckling experiments that I explained to study the forces, but we can now do that also in combination with control molecules. So this is just a recent example of that pushing force generation experiment that we've been doing for many years, but now with control molecules, and we can really in a lot of detail, when a growing filament hits a boundary, which in this case is a glass wall, and it's decorated with this hitchhiking molecule shown in green, and this is a blow up of that. In a lot of detail, we can really correlate the dynamic properties of the filament to what these control molecules are doing at their end. So it provides us with a very quantitative way to look at the combined effects of molecular control and force, which are both relevant in real cells. So what about minimal spindles? So we can study how individual esters position themselves, but real spindles, of course, are more complex in many ways, and we're going step by step. First of all, as a real spindle, of course, has two estral structures. It has chromosomes in the middles, and every cell biologist can tell you about many other components that we know are very essential to properly organize the real spindle. But so to get there, I'm now showing a bit of the technical details. We use these microfluidic techniques, increasingly actually also to make vesicles like other people do, but what I'm showing today is in droplets. So I don't know if many of you use the same technique, but you use a PDMS device where, as you see on the bottom, we simply push a buffer, water containing fluid with all the proteins and other components that we need inside our droplets from the left into an oil phase that has lipids inside, and then you create bubbles or containers with a monolayer of lipids around it. And unlike cells or vesicles, these are quite hard containers. The surface tension is quite high. So it's not very deformable. It really provides us, unless you shape it in some other shape proactively, you get spherical containers, and we can functionalize the lipids with things like biotin so that we can hook up molecules there. And for these positioning experiments, for example, we would put dynein molecular motors on these lipids, then put in a microtubule nucleating structure called the centrosome. It's isolated from cells, put enough protein and energy and other things to make sure that bleaching doesn't happen and ATP doesn't run out. There's lots of extra things that you don't see that are needed for successful experiments. And then ask, depending on the length of these filaments on average, depending how much force pushing versus pulling can be generated at the edge, what do you expect for the positioning behavior? And this is old work that was done together with theory from Frank Jülicher's group. And we showed that if you have a single ester and nothing can pull on the microtubule ends, but the microtubules can only grow and push, then eventually these esters will always be destabilized, we call it, going to the edge of the cell. Whereas if you put a molecular motor on the edge that can also pull, then when the filaments are long enough, and then pulling is always needed to get to this faithful positioning. And you can show that this depends, how efficient this pooling can operate depends on the geometry. It works well in 2D and 3D, but not in elongated structures, for example. And then you can also add 2 of these centrosomes to get closer to a real spindle. And then we found that if you have two esters with long filaments that reach the boundaries of these containers, that not only do they push individually to the side of the container, and this is just the energetically most favorable solution if you take into account the bending properties of these filaments, but they also repel each other due to steric hindrance. So you get already this bipolar organization where they sit on opposite sides of the bubble. And then if you combine these slight repulsive forces, which you can also predict using simulations again, what the magnitude should be, and this is a collaboration with Bela Mulder from Amsterdam. You combine this repulsive force with a centering force, as we call it, due to molecular motors, then you get quite robustly a situation where there's a force balance between a centering force due to pulling forces at the periphery and a repulsive force due to steric hindrance between the filaments that puts you in a sort of intermediate steady state situation where neither the centrosomes are both in the middle nor are they at the edge. So and just to, because we can compare to models, we also understand why this is quite robust and not so dependent, for example, on how many filaments are able to grow or how long they are exactly. So that's the strength of these simple experiments. Of course, we also know that in real spindles, there's chromosomes in the middle and there's molecules that that crosslink microtubules coming from both sides. So in this particular example, we've added the crosslinking molecule in the middle. That's why you see this bundle of filaments spanning the two esters. And we've shown that that doesn't do much extra to the intrinsic organizational behavior of these two esters, except if you add a lot of this crosslinking molecule, because then you get an extra force that actually pulls these two centrosomes together, and you do not get this bipolar organization anymore. And we've tried a few other things. I'm not going to go through all of it, but the message being that if you have this minimal spindle that is completely naive, but where you at least understand the force balance, you can add other components that we know are important in real cells and ask, what do they change? And then sometimes you find you add one component, nothing changes. But you need to add it in the context of a second or a third component, and then it does change something. And that's, of course, how cells also work. It's always redundant. There's also always combinations of molecules with the same effect. And this is sort of a playing ground for bottom-up understanding what the effect of these combinations of molecules is. So in addition to understanding the force balance in this steady state situation, we would like to also understand a bit more about, not only the static situation or the steady state solution to this problem, but also worry about the dynamic properties. As I showed in the beginning, C. elegans embryos actually position their spindle a bit asymmetrically. So what does it take to get a robust asymmetric positioning? And then can we also bring in mimics of chromosomes or pieces of DNA and ask, what does it take to organize these things in the middle? And can we eventually combine with some kind of oscillatory temporal control to also separate chromosomes? And this is where we are already heading in the direction of this synthetic cell projects, where we want to use this structure now also to separate pieces of DNA that we can maybe then use combination with other modules in this larger project with synthetic cells. So two examples of how we now try to do this. So introducing minimal chromosomes is something that we've worked on in collaboration with the group of Andrea Musacchio. So he's a structural biologist from Dortmund, specializing in reconstituting the mammalian kinetochores. So kinetochores are protein complexes that hook up chromosomes to dynamic microtubules, and he also makes very well controlled, biochemically perfect, I would say, minimal versions of this with his postdoc Pim Huijsens-Veld. And we would like to put eventually these complexes that are shown here to really faithfully track shrinking microtubules, to put them on beads and then study the self-organization behavior in these minimal spindles. This is a project by Ali Nikmalevi, who just recently started in my group. And then we also want to study this asymmetric positioning. And this is a project we've been struggling with now, a technical thing that we've been struggling with for years, but getting a bit of optimistic feeling that it may work one day. And the idea was that we make these microfluidic droplets asymmetric in shape, so elongated, and they use light-induced protein recruitment, so optocontrol, if you want, to recruit molecular motors to the periphery of these droplets, not in a symmetric way, but an asymmetric way, because that's how it seems to be controlled in cells. So you can use molecular motors that you hook up to the lipid interface with a dimer system that responds to light. And this is just showing that if you do that on a supported lipid bilayer, that easily works. But inside the microfluidic droplet, it's much harder to make it to work. But the idea being that we can shine light where we want the motors to stick to the interface, and then we turn off the light, they let go, and this way control where molecular motors are able to generate forces. So that's two examples of work in progress. So I want to have some time for questions, so I have a few slides about the polarity example, but I think it's a similar spirit, so I'd rather skip those and go directly say a little bit more about this larger effort of building synthetic cells. I use the last 5 minutes for that, if that's okay. Yeah. So if you want to know more about this polarity, I knew this was going to happen, but so we reconstitute using similar spirit, also the ability to polarize membranes, and that's what this was supposed to show. But I'll leave it for my next visit or questions afterwards, and really go, what does this now bring to this larger consortium? So as I explained in the beginning already, quite extensively, I think it's trying to build minimal versions of cells should teach us a lot about how real cells work, but you may also have noticed that our efforts to build cytoskeletal structures are now going on for 10, 20 years already, so it's also slow going. So, I think now there's enough known how to do this in vitro by many groups that the timing is right, but there's just no way that anybody's ever going to be able to do this alone. So it really requires a lot of collaborative efforts. And that's why also we started this community in Europe, where we say, okay, there's lots of ideas. It doesn't even, if you build a cell-like system, it can be based on known biochemistry, but you could also take supramolecular chemistry or alternative approaches or DNA origami. There's all kinds of approaches you could take in building lifelike systems. So this and bringing this community together could be really exciting. In the Dutch context, we build a consortium that really has a starting point biochemistry as we know it. So we say, okay, that can we build a minimal mimic of a simple cell, as I already explained in the beginning. So we can contribute on the cytoskeletal or cell DNA separation side maybe. But even if you want to keep it simple, there's lots you have to worry about. If you want to be serious about autonomy, somehow you need to produce energy, maybe not its variety as real cells, but at least some version of it. We have to replicate DNA if we want it to be DNA based and have proteins expressed. And worry about the container growth, let's say. You need a membrane that can grow by adding lipids. Somehow needs to take care of its shape, control its shape, keep it robust, and eventually divide. So we have a 10-year program, and it's really, we're very thankful that even though funding is always very difficult to get, it's very competitive, at least we have a program in the Netherlands that allows you to get together with, in this case, 17 groups from 6 different universities to work for 10 years on a goal. So that's great. What we said is, okay, in principle, we know the genetic code for all cells, but as I explained in the beginning, we can even maybe know all the molecules that are eventually involved, but how does life emerge from interactions between them? And what is the minimal set of components that you need? And there, like I already explained, you could take either bottom-up or top-down approach. And our proposition is that you need at least 3 things. You need to somehow take care of metabolism, what we call self-fueling here, making small, complicated molecules out of simple food. You need to process DNA, replicate and translate, and then you need the mechanical side, which is division. So we put together, like I said, a group of experts that are each over the last years have been working on parts of this. So we have biochemists from Groningen who can have sustained ATP production in vesicles. My colleague Christophe Danelon in Delft is an expert in vitro gene expression. So you have vesicles with DNA and then a mix of molecules that allows you to express proteins from this. And then eventually we want to, first using purified components, learn enough about all these different modules and learn how to integrate them. And of course, you know, there's lots of challenges. I don't have to convince most people that it's difficult, but we're going to try anyway. And we need modeling. So there's these modules that we need to learn how to integrate. And modeling is very important to help guide us. And then we also have a top-down side to this project. So we have John van der Oost and Pascal Dahan. They are really microbiologists who are experts in genome engineering, so that we can also top-down approaches sort of in our program to check what we are doing, maybe replace modules that we've designed in minimal cells and see if they can adapt to the modules that are present in a real cell. And then also interestingly, a professor who is an expert in philosophy and ethics, because, you know, are we creating life or should we be doing this at all? So that's also a very interesting side to this story. And maybe just as one technical last slide, I've been, you know, our expertise is in eukaryotic microtubules, but we are more or less convinced that for this minimal cell, we may need to go to a simpler cytoskeletal filament that, for example, can be expressed. So these in vitro expression systems that, oops, sorry. And this is an automatic slide. That's a problem now. Yeah, so these in vitro gene expression systems, they work well with prokaryotic proteins and not at all with this eukaryotic tubulin that we've been working with so far. So that's why in my group, we've been in the last 2 years starting up pilot experiments where we now study the biophysical properties of alternative cytoskeletal polymers that are actually simpler than the eukaryotic ones and found in bacteria. So that will then allow us to express these polymers in a vesicle and use them to deform, elongate, and maybe separate pieces of DNA eventually in a way that is simpler than the spindles that I've been showing you so far. So I want to stop and thank the many people that over the years we collaborate with. It's always an effort. You know, we are experimental biophysicists, but like I said, this really depends on collaborations with cell biologists, structural biologists, theory people, and many of them are shown here. Also people providing us with all kinds of materials and advice, funding agencies, and of course, most importantly, the people in my group. So this was a picture taken when we were still allowed to get together, walking on the bottom of the sea in the north of the Netherlands. So if you ever want to visit and walk on the bottom of the sea with us, you're very welcome. This is where I'll stop.
Okay, thank you very much for the great talk, Marileen. It was very inspiring. As a researcher working in the field of synthetic biology, I'm really amazed by the synthetic cell building effort. So this was very inspiring again. Thanks very much. Thank you very much. So the talk is open for questions, dear audience. So if you have any question, please just unmute yourself and ask your question.
Should I stop sharing or?
Doesn't matter.
It doesn't matter. Okay, maybe there's still slides too.
Any questions?
Let's see.
There is one question from the chat screen. So
okay.
It says, I may have missed during the talk, but have you used recombinant proteins as well?
Yes. Yeah, so we, so it's a mix. So the tubulin, for example, that we use is isolated from brain tissue, sometimes by us, sometimes by a company that we buy it from. Mm-hmm. But the molecular motors, dynein, for example, is a motor that we express in yeast cells, and it's not the wild type full-length dynein, but it's a shortened version with a GST tag so that we can dimerize it artificially and then biotinylate. We have also non-existing proteins that we use sometimes. I didn't show it here, but we also are interested in looking at crosstalk between microtubule and actin cytoskeleton, together with my colleague Gijsje Koendrink. And then we make microtubule actin crosslinkers that do not exist, but we take pieces of real proteins that we glue together with an artificial double helix. So there's a lot of protein engineering depending on the experiment that we do. And we also mix with the microtubule cytoskeleton. You can do that. It's very evolutionary conserved. So if you take a tubulin from a pig brain or a cow brain or any other, or plant cells even, you can easily combine it with proteins coming from yeast, because it all, not always of course, but it interacts with each other quite faithfully. So it's really a mix, yeah. And the only one that we get from tissue is tubulin. The rest is all expressed in cells in our lab, yeah.
Okay, any other questions?
I may ask one more question.
Go ahead, Elif. Please, Elif, ask your question.
Okay. Which components do you think is required for an artificial cell? For example, just, you can hear me?
Yes, yeah. I'm just
okay.
about changing my screen that I can see some more of you. I don't know.
Okay. Okay. Do you think that a nucleus is required for an artificial cell, or
a nucleus?
Which components are required for an artificial synthetic cell?
Yeah, so it depends a little bit on how you define the end goal. So in our consortium, we said we're aiming for a system that can take care of its own energy production. So you need enzymatic systems that can generate or synthesize ATP. Doesn't need to be ATP synthase. It can be in a bacterial set of enzymes that is able to make ATP. We said we need molecular systems to replicate DNA and then also control molecules to control the timing and translate into protein. And then we need molecular systems that allow that will produce lipids so that you can have a growing container. And then eventually in mechanisms that we really haven't decided on yet what is the best to separate the DNA and constrict the cell. So we've defined it in terms of minimal functions that we think are needed. What that exactly means for the set of molecular components that is required to execute these functions together in the same system, that's, I guess, what we're going through. So we're trying out different strategies, but the minimal functions we want is a minimal form of metabolism, DNA replication, translation, and growth and division. So those are, and that's people might say, well, you know, to call it a minimal living cell or a synthetic cell, it also needs to communicate with the outside world and be able to evolve. So that's I think a valid argument that we have not put on our wish list for this project. But so then I think we will get to a system that can do maybe some of the things that we would like, but not all. And then, you know, have you built a synthetic cell, or you know, when is it done? I think that will change over time. But this is at least the goal we set ourselves. And the number of components, you could also ask how many identical or individual, you know, how many different kinds of proteins would you need? And we think about 200. Plus or minus 100. And the reason is that, for example, the Greg Venter Institute, when they do the top-down approach with these simple bacterial cells, they could reduce the genome to a few hundred, like around 400 genes. And then the cell would still be alive, you know, not great. But so apparently a few hundred is enough in the top-down approach, which makes us think that with several hundred genes in our bottom-up system is the order of magnitude that we are thinking about. But what exactly? I think in the Greg Venter minimal cell, the one they published in 2016, I think there were 473 genes. Of course, they've progressed since then, but 30% of them was unknown what they were for. So that's very interesting. So maybe are they needed and we just don't know why? Or if you do the experiment again, of course, top down or bottom up as well, actually, you may easily get in a local minimum of what works and another set of, sometimes you take away a component, it doesn't work anymore. You take away another component and then it works again because you have antagonistic effects. So, but yeah, if that's the question, I think a few hundred identical components and different components, different genes is what we are counting on. Yeah. I hope that answers your question.
Okay, thank you very much.
There are some more questions, Marileen. One is, do you also, Merve Nida is asking, do you also focus on reconstituting actin and intermediate filaments?
Yes, so but this is a collaboration with Gijsje Koendering. So she's a professor also in Delft. She was in Amsterdam for a long time, but recently joined our department, and she's taking a similar approach, biophysical reconstitution approach to the actin cytoskeleton and intermediate filaments. So it's really her expertise, and together we're also combining these. So we have we would like to look at the self-organization of these spindles also in systems where we introduce a cortical actin cytoskeleton and look at the crosstalk between the two. And it's known, for example, that crosstalk between microtubule and actin cytoskeletons helps the cell decide where to constrict. So a cytokinetic ring, and again, back to the eukaryotic examples, which there's always this double interest, trying to understand eukaryotic complex cells better, and then learn from these approaches to build minimal versions for the synthetic cells, which are not necessarily exactly the same experiments you would do. But in the context of understanding eukaryotic cells better, we know that the mitotic spindle, with the help of molecules that live at the ends of microtubules, interacts with the cytokinetic ring in the middle of the cell, and the positioning of the cytokinetic ring is dependent on signaling that happens in crosstalk between these two cytoskeletal systems. So this is the kind of crosstalk we are trying to reconstitute in droplets, but also in vesicles together with her. Yeah, and this is actin microtubule. Actin plus intermediate filaments is something that Gijsje is interested in, that I'm not involved in. So septins and intermediate filaments that interact with membranes is also her interest, but not something we work on.
Okay, thank you very much for your kind answers and for your presentation again. With this, I just want to finalize the colloquium for today. So once again, we hopefully want to see you over here in person as well very soon. I hope so. So yeah, thank you very much again, and have a great weekend. And I also wish everyone a great weekend. Thanks for being here and listening to this wonderful presentation.
Okay, thank you again, and hopefully see you soon.
Sure, sure.
For real. Yeah. Bye-bye.